This is the third video in my series on antimicrobial drugs. In this video, we'll look at inhibitors of translation, we'll look at inhibitors of nucleic acid synthesis, and we'll look at inhibitors of essential metabolite synthesis. Your main objective for this video is to make a list of the different antibiotics discussed and make a little note of how they inhibit the growth of microorganisms. You'll also want to make a note of the different methods that bacteria use to avoid harm from antibiotics and antimicrobials. These notes may be useful to you as you practice and get ready for exam number 4 and your final.
This lecture goes with Chapter 20 in Professor Foster's Elements of Microbiology at Bristol Community College. So we've been talking about antibiotics, and we started out by introducing some vocabulary. We talked about terms like broad spectrum versus narrow spectrum. We talked about selective toxicity. We even started talking about antibiotics. We started with those antibiotics that target cell wall synthesis. So remember talking about penicillin. We probably talked about bacitracin, vancomycin. We may have talked about cephalosporin. And we said that those types of drugs that target cell wall synthesis, they are called selectively toxic because they target peptidoglycan, which is a molecule that you only find in bacteria. Therefore, unless someone or a host organism is allergic to penicillin, that drug only affects the bacterium and not the host, normally, unless someone's allergic. Now we want to look at drugs that target another process that you find in bacterial cells, and that's protein translation. And you'll probably remember that protein translation is an essential function inside of the cell. Proteins, remember we've been calling them the workhorses of the cell. They're responsible for moving molecules into the cell and out of the cell. They transport substances from one location to another. They move cells from here to there. They're responsible for replicating DNA, for metabolizing nutrients and converting them into energy. Enzymes do so many different things in the cell. A cell cannot live without proteins. So protein translation is the term that we use when we really mean protein synthesis. And you'll hopefully remember from a previous biology class that protein synthesis happens at the ribosome. So this kind of red structure that you see on the slide here is a ribosome. Remember that your ribosomes in bacteria are made up of two subunits. You have a large 50S subunit that looks kind of like a gumdrop in this figure. And then you have a 30S smaller subunit that the gumdrop sits on top of. And these two pieces assemble onto messenger RNAs. So remember, messenger RNA is basically the RNA copy of a DNA gene, and it encodes the amino acid sequence for making protein. So this is kind of like having the blueprint for making a protein. Now, there's a third player that's necessary for us to make protein. So we have the ribosome and messenger RNA. What we're missing is tRNA, or transfer RNAs. So remember, transfer RNAs bring amino acids basically to the ribosome, and then you have a chemical reaction that happens in the ribosome that joins these amino acids together via peptide bonds to make proteins. Now, if you need a review or a refresher on how this process works, you'll probably want to follow the link that's on this slide, or follow the link that I've posted to your Blackboard web space. It'll go through the process with you, and it may be useful to review so that you're familiar with some of the terms that I'm going to use through this presentation. Now, when it comes to antibiotics that inhibit protein synthesis, there are three main steps that are going to be affected. Okay? The first step that's affected by antibiotics is the formation of what's called the 30S initiation complex. So basically, right before protein synthesis starts, you have the assembly of messenger RNA on that small 30S subunit of the ribosome. Okay? You'll also have a transfer RNA that contains an amino acid, the methionine amino acid, attached here. So this process, the assembly of these three structures, can be inhibited by antibiotics. Okay? So for example, this is what your tetracyclines do, and we'll talk about that in a little bit. Another step that can be inhibited during protein synthesis is the formation of the 70S ribosome. So when you have that 30S initiation complex assemble with the 50S portion of the ribosome, okay, protein synthesis can begin, and this particular step can also be disrupted by antibiotics. Okay. A third step that tends to be inhibited is the formation of protein. So there are antibiotics that are actually going to bind in the 50S portion of the ribosome, and they're going to prevent peptide bond formation between the tRNA, or the amino acid attached to the tRNA in the A site, and the growing polypeptide chain that you find in the P site. So let's talk about some antibiotics. We're going to start with chloramphenicol. Now, chloramphenicol is not prescribed so much in the U.S. Anymore, but it is fairly common in the developing world. So I'm going to mention it here, but we won't spend a lot of time on it. Chloramphenicol is a broad-spectrum antibiotic. So remember, we said that broad-spectrum antibiotics treat not just one type of bacterial cell, but they treat many different types. So broad-spectrum here means gram-negative and some gram-positives. Chloramphenicol basically binds in the 50S subunit of the ribosome, and it prevents peptide bond formation between the tRNA and the A site, so that amino acid that's attached to that tRNA in the A site, and the growing polypeptide chain that you find in the P site. This drug is a bacteriostatic drug. Our next set of drugs you do find prescribed here in the U.S., and you'll actually find some of them on the drugs in your drugstore on the first aid aisle. So these are your aminoglycosides. Your aminoglycosides consist of two amino sugars, basically, that are linked by glycosidic linkages. Okay. So you can see kind of an example of this on the right side of the slide. Don't worry, I'm not going to expect you to remember this structure. It's just there because I think it's kind of interesting, and I thought you might like to see what this drug looks like. So your aminoglycosides are broad-spectrum antibiotics. They're going to treat gram-positives as well as gram-negatives, and this is one of the few protein synthesis inhibitors that's actually bactericidal. Remember, I told you there's not many of them. This is one. Usually your aminoglycosides are used to treat life-threatening infections, and they're often prescribed as part of a cocktail, a drug cocktail, because there's quite a bit of resistance to these drugs already, and when they're part of a drug cocktail, the idea is that more resistance won't be developed, or it shouldn't be as easy for the bacterium that you're treating to develop resistance. Now, one of the cautions in using these drugs is that the therapeutic dose, that is how much of the drug you have to give in order to kill bacteria, is close to the toxic dose, that is how much antibiotic the host organism can take before getting ill. So those two doses are very, very close, which is one problem, and some might consider it a disadvantage when using these drugs. Your aminoglycosides include drugs like your streptomycins, your neomycin, gentamicin, and tobramycin. Another aminoglycoside that you should be aware of is amikacin. Amikacin is a last line of defense drug, so that means that when you have a bacterium that's resistant to many different antibiotics, this is the drug that you use to try to kill that multi-drug resistant bacterium. So amikacin is used to treat multi-drug resistant gram-negative bacteria. So what do these antibiotics do? Your aminoglycosides change the shape of the 30S subunit. This interferes with the formation of that 30S initiation complex that I talked about. Also, this interference causes the messenger RNAs to be misread. Okay. Another function that's purely seen in the gram-negatives is that treatment with these aminoglycosides can cause the outer membrane of your gram-negatives to leak. I don't know the mechanism for this. I'm not quite sure why this happens. I'm not sure that anybody really understands exactly why this happens yet, but you should be aware of it. Now, when it comes to your aminoglycosides, remember I told you that the therapeutic dose is really close to the toxic dose? Well, it turns out that there are a lot of side effects when you actually get close to that toxic dose. So you'll get you could find in your patients renal failure, so that's kidney failure, vestibular and cochlear toxicity. So remember, the vestibular system and the cochlea are responsible for equilibrium and balance. So people on these drugs for an extended period of time, or people who are having high doses of these drugs, may have problems with equilibrium and balance. There's also auditory toxicity. So there's some damage, for example, to the auditory nerve. Okay. Now again, with most normal treatments that last for under 2 weeks, people won't experience these severe side effects. But again, when someone's on a high dose for, say, a short period of time, or they're on these drugs for more than 2 weeks, these are some side effects that can be expected. Now, one thing that we kind of looked at or mentioned when we were talking about cell wall, when we were talking about drugs that target the cell wall, is resistance. So many bacteria have mechanisms in them to resist the effects of antibiotics. Back when we were talking about the cell wall, we mentioned that some bacteria have enzymes like beta-lactamases that break down the beta-lactam ring, thus inactivating the enzyme. Remember that? We also said that some bacteria have transpeptidase enzymes that are mutated so that the binding site for penicillin has changed and penicillin can't bind. Well, it turns out that some bacteria have resistance against your aminoglycosides. This resistance typically is through enzymes that are going to modify the aminoglycoside so that it becomes inactive. All right, so maybe they phosphorylate the drug, for example. Then it can't bind where it's supposed to, and it's inactive. The genes for these inactivating enzymes are passed on plasmids. So we've already talked about how genetic information passes on plasmids. You can pass information via conjugation or transformation, for example. Next, we have your tetracyclines, and I suspect that all of you have already heard of tetracyclines before. These drugs are broad spectrum drugs, so they treat gram-positives, gram-negatives. They also will treat intracellular parasites, so bacteria that live inside of the host cell. Tetracyclines are bacteriostatic, so that means that these drugs are going to keep bacteria from growing in hopes that either another drug will kill the bacteria or the immune system will kill the bacteria. Some cases in which tetracyclines get used At low doses, tetracycline might be used to treat cases of acne or bad acne. It can be used to treat chlamydia infections, mycoplasma infections, brucella infections, and helicobacter infections. So helicobacter, you might remember, is a bacterium that is associated with stomach ulcers and to a lesser extent stomach cancer. So this drug would kill those bacteria. Most of the time, tetracyclines are not the first line of defense drugs against these bacteria. Usually it's second or third line. But in the case of Borrelia burgdorferi, the bacterium that causes Lyme disease, and in the case of Ehrlichia, which is an intracellular parasite that's transmitted by ticks, and it is bacterial, this is actually the first drug of choice. So interestingly, your tetracyclines don't, as far as we know, they don't penetrate mammalian cells, but they do appear to kill intracellular pathogens, again, like Ehrlichia. So this antibiotic can be used for treating that type of bacterium. Some examples of your tetracyclines include, well, tetracycline, doxycycline you might have heard of. That's used sometimes for treating MRSA, and minocycline. And one of the disadvantages to using tetracycline for treating any kind of infection is that a lot of bacteria actually have mechanisms to resist this drug. Before I leave this slide, I want to draw your attention to the right hand lower corner. This is tetracycline, and your tetracycline drugs will all have that structure where you have the 4 fused rings. It's just that the chemistry around those rings will change. So your tetracyclines work by binding the 30S subunit, that's that small subunit of the ribosome, and they prevent the attachment of charged tRNAs to the ribosome. So what's a charged tRNA? These are basically tRNAs or transfer RNAs that have amino acids attached. So during protein synthesis, these charged tRNAs diffuse into the A site of the ribosome, and then if the anticodon on the tRNA matches or is complementary to the codon on the messenger RNA, you'll get a chemical reaction that results in the synthesis of a peptide bond between the amino acid on the tRNA in the A site and the growing polypeptide chain in the P site. So tetracyclines keep that from happening. Some side effects that people experience when taking tetracycline include photosensitivity. Children who are on this drug may have some gray discoloration on their teeth. Some people experience hepatotoxicity, and then some people may be hypersensitive to this drug. As I mentioned on the first slide, lots of bacteria have some level of resistance to your tetracyclines. This resistance happens through efflux pumps. Okay, so what an efflux pump is a protein that sits in the plasma membrane or in the cell wall, and as antibiotics kind of cross into the bacterial cell, these pumps pump that antibiotic right back out. So this keeps the concentration of antibiotic in the cell relatively low. The genes for these efflux pumps are encoded on plasmids, and these plasmids can be passed from one bacterial cell to another. The unfortunate thing about these efflux pumps is that they confer resistance to all tetracyclines. So it doesn't matter whether someone is on doxycycline, minocycline, or tetracycline. If the bacterium that's causing their infection has this efflux pump, that bacterium will be resistant to all of those tetracyclines. Streptogramins are the next group of drugs. These are effective against gram-positives only, so they have a very limited spectrum of activity. This is another group of drugs that are bactericidal. Okay, so again, very few antibiotics that inhibit protein synthesis are bactericidal, but this is one of them. What you need to know about your streptogramins is that they are used primarily to treat vancomycin-resistant bacteria, especially your vancomycin-resistant enterococci and vancomycin-resistant Staph aureus. Okay. Now, here's the kind of cool thing about this drug. It's actually not just one drug, but it's two. So the drug is Sinercid, and it's made up of two bacteriostatic peptides. So the first one is quinupristin, and the second is dalfopristin. And these two peptides have what's called a synergistic effect. So synergism basically means that when we give both of these peptides together, they have a stronger effect than if we were to administer only one. Okay? So as I told you, the quinupristin and the dalfopristin are both bacteriostatic. When we add them together into the same drug, they have a bactericidal effect. So again, that's a bigger effect when we give these two together versus when we administer these drugs independently of one another. The way that these work is that they bind the 50S subunit, and they have two effects there. The quinupristin is going to inhibit elongation of growing polypeptide or growing protein chains from the ribosome, and the dalfopristin is going to prevent peptide bond formation from the tRNA in the A site to the growing polypeptide chain in the P site. So both of these things together stop protein synthesis, and they lead to death of the cell. Some of the some of the side effects of your streptogramins include muscle and joint pain and phlebitis at the site of injection for this drug. Some bacteria, unfortunately, are resistant to your streptogramins. Bacterial resistance is basically conferred through methylation of the ribosome, so a methyl group is added to the binding site for these drugs on the ribosome. That changes the shape of the site and prevents the drugs from binding. Okay? And then you also have efflux pumps. So again, efflux pumps sit in the cell, and basically as antibiotic comes into the cell, these pumps pump the antibiotic right back out to keep the concentration of drug inside of the cell low.
Let's take a break really quickly and see what you remember. True or false? Aminoglycosides are considered to be magic bullet drugs because they only harm the microbe, never the host. The answer for this question is false. Your aminoglycosides have several side effects, particularly when they're used at higher doses or with frequent doses. Here's another one. Antibiotics that are synergistic are blank when given together versus when given alone. Is it antagonistic, more potent, or less potent? Right. These drugs are more potent when they're given together. An example is Sinercid. Remember, Sinercid is made up of two peptides. When given independently, they're simply bacteriostatic, but when given together, bactericidal. How about one more? Which of the following is a broad-spectrum antibiotic? Is that tetracycline, vancomycin, penicillin, or streptogramin? Right, tetracycline. Tetracycline is effective against gram-positives, gram-negatives, and intracellular parasites.
So this next group of drugs are kind of one of my favorites. These are the macrolides. And the reason I like the macrolides really is based on their shape. So if you look in the bottom right of this slide, you'll see that the antibiotic is really kind of large, but it consists of this star-shaped component called a macrocyclic lactone ring. And I think that's just a really uniquely shaped structure, which is, I guess, why I like this antibiotic. Anyways, you have different types of macrolides. They're divided up into first and second generation drugs. So erythromycin is your first generation drug, and it treats only gram-positive bacteria. This is, your macrolides are bacteriostatic, so they just inhibit the growth of bacteria. They don't actually kill. Okay? So again, your erythromycin, it's only against your gram-positive bacteria. And this is the first generation macrolide. Your second generation macrolides are called azolides, and they have an extended spectrum. So they're going to kill not only gram-positives, but they also kill some gram-negatives. And I bet you are familiar with one of these already. So azithromycin, if you've ever had a Z-Pack, this is the drug that's in that Z-Pack. It is a macrolide. Okay, it's one of the second generation macrolides, and again, it has an extended spectrum. And clarithromycin is another second generation drug. Your erythromycin, azithromycin, and clarithromycin all are designed to treat community-acquired infections. So that means these are going to be infections that people get just from being out and about. Okay, they're not acquired in a healthcare type of setting. And I will tell you that macrolides do treat a variety of different infections. The way that your macrolides work is they bind the 50S ribosome subunit irreversibly, and they block movement of the ribosome along the messenger RNA. Okay? Now, the reason why erythromycin is effective only on your gram-positives is it's so large it's not able to penetrate the gram-negative cell wall. Remember, your gram-negative cell wall has not just peptidoglycan, but it has that outer membrane. And embedded in the outer membrane, you have these porin proteins that make a hole so molecules can come into and out of the cell. Well, that porin is so small that the macrolide antibiotic can't actually fit through it. So that's why your erythromycin is gram-positive only. When we get to your second generation macrolides, or your azolides, the antibiotic structure has been changed to kind of overcome this limitation. Generally, your macrolides and your azolides are very well tolerated, so you don't have a lot of side effects from these drugs, but sometimes people will have problems in their gastrointestinal tract, so like cramping, diarrhea, things like that. Some bacteria do have resistance to your macrolide antibiotics. They get it through methylating the 50S ribosomal subunit. Adding methyl group to this subunit changes the shape of the binding site for this antibiotic and prevents the antibiotic from actually binding the ribosome and taking effect. You also have efflux pumps that pump the antibiotic back out of the cell once it enters. Okay. There are also a few inactivating enzymes that essentially destroy the structure of the antibiotic or modify the structure of the antibiotic so that it cannot bind the ribosome properly and it's not effective. I've posted a link to a video that I like about your macrolide antibiotics to your course web space. If you want to look at this topic again, I recommend taking a look at that video. Lincosamides are kind of related to your macrolides in function. They bind the 50S ribosome, and they're going to cause premature ejection of tRNAs in the P site. This is going to stop essentially protein translation. Now, your lincosamides are used primarily for treating problems caused by aerobes or staphylococcus, particularly toxic shock syndrome. Sometimes they're prescribed for infections by streptococcus bacteria, And it's important to know that these drugs are bacteriostatic, just like your macrolides. Two examples of lincosamides are clindamycin and lincomycin. Oxazolidinones are the next group, and what's special about this particular group is that it really consists of just one drug, and it's a completely synthetic drug. Linezolid was developed in a lab, basically, with the idea that it would be harder for bacteria to have resistance to this drug because it doesn't occur naturally in the environment. The bacteria that you might use the bacteria that you might treat with your oxazolidinones haven't had thousands of years to develop resistance to this drug. Okay. The linezolid was developed to treat vancomycin-resistant bacteria. It was also is basically an anti-MRSA drug. Your linezolid treats gram-positive infections, okay? And what the drug does is it binds the 50S subunit of the ribosome and prevents that from assembling with the 30S to form the 70S. Your side effects from linezolid include thrombocytopenia. So this is a deficiency, basically, in your platelets. Okay, and myelosuppression is another side effect. So myelosuppression suppresses the formation of blood cells, both your white blood cells and your red blood cells, and it suppresses those cells that eventually become platelets as well. We have seen some resistance, although not a lot, to your oxazolidinones. Bacterial resistance occurs through mutations at the binding site for the drug. Those mutations change the shape of the binding site, and then the drug can't bind there, and it loses its effectiveness.
Here's another practice question. What do you remember? Macrolide antibiotics like azithromycin, remember that's your Z-Pak, work by, is it number 1, binding the 50S subunit of a ribosome and preventing translocation? Is it binding the E site of the ribosome, causing premature termination of protein synthesis? Or is it binding the 30S subunit and preventing assembly of ribosomes? What do you think? That's right. Binding the 50S subunit of a ribosome and preventing translocation is exactly how macrolide antibiotics like Z-Paks work.
Now we're going to move away from those drugs that target protein synthesis, and we're going to go to drugs that target the plasma membrane. So we have one drug for this class, and that's polymyxin B. This is a drug that is only used topically, so it's in ointments and creams. It's combined with bacitracin and neomycin and neosporin. And this particular drug is effective against your gram negatives. So what this means is in neosporin you have one drug that's effective against your gram positives, that's the bacitracin. You have one drug that's against your gram negatives, that's the polymyxin B, and then you have a drug that's effective against both, which is your neomycin. Some drugs that we use as antibiotics are going to inhibit nucleic acid synthesis. So two examples of this include your rifamycin, which inhibits messenger RNA synthesis by binding your RNA polymerase and inactivating it. Typically, rifamycin is used as an anti-tuberculosis drug and an anti-leprosy drug. And it has this really quirky characteristic that people who are on this drug usually cry orange tears and their urine turns really orange. Usually rifamycin, when it's used as an anti-tuberculosis or an anti-leprosy drug, it's administered with other drugs as part of a cocktail. So, for example, someone might be on ethambutol and rifamycin to treat their tuberculosis. Your quinolones and your fluoroquinolones are another class of drugs, but their target is actually DNA synthesis. Okay, so rifamycin, that's messenger RNA synthesis. Quinolones and fluoroquinolones, they target DNA synthesis. A common drug that belongs to this group is nalidixic acid. It gets prescribed to treat urinary tract infections. It's a fairly common drug that gets prescribed. Maybe you've heard of ciprofloxacin, also called Cipro, belongs to this group of drugs. And basically what these drugs are going to do is inhibit DNA gyrase and topoisomerase. Okay? An important thing to note about these drugs is that they're targeting bacterial enzymes, not eukaryotic enzymes. And for this reason, these drugs can be used to treat bacterial infections. I have posted a link in your course web space for these two antibiotics. You should take a look at that video. It talks about them in a little bit more detail. It also talks about resistance to these drugs. The last group of drugs that we want to talk about with respect to bacteria are your competitive inhibitors. So if you look at this figure, you'll see a kind of green blob on this slide. That represents an enzyme. And the active site for the enzyme is at the top of this green blob. And the substrate that this enzyme usually binds is represented by the red blob. Okay, so you can kind of see how that might fit into the active site on the green blob. If it weren't for this little yellow blob, that's our competitive inhibitor molecule, the red blob could fit into that active site. What competitive inhibitors do is they bind in the active site of enzymes, blocking the active site so that the normal substrate can't bind. So that's what you're seeing here. Your sulfonamides, aka your sulfa drugs, do precisely this. Your sulfonamides were among the first synthetic antibiotics developed, and we want to look at one that is structurally similar to PABA. So PABA stands for para-aminobenzoic acid, and it is a precursor to folic acid. So many bacteria actually make their own folic acid, and they need that folic acid so that they can synthesize nucleotides for making DNA and RNA. So normally what happens is you get a pathway that looks kind of like what you see in the middle of this slide. So the PABA is converted into a molecule called dihydrofolic acid by an enzyme called dihydropteroate synthase. Then that dihydrofolic acid is converted into another molecule called tetrahydrofolic acid by another enzyme called dihydrofolate reductase. And then that tetrahydrofolic acid can be used to make nucleotides. Okay? Now, sulfamethoxazole is a sulfa drug, and it acts as a competitive inhibitor for PABA. So sulfamethoxazole can bind to the dihydroteroate synthase and prevent the formation of the dihydrofolic acid. This interrupts that pathway for folic acid synthesis. Trimethoprim is another drug that is sometimes given with sulfamethoxazole. In fact, if you've ever had Bactrim, then you've taken both of these drugs together. Now remember, in synergism, you get a greater effect from two drugs that are administered together than you get from administering either drug by itself. So what trimethoprim does is it acts as a competitive inhibitor for dihydrofolate reductase. So that drug binds the enzyme instead of dihydrofolic acid. And this again disrupts the production of tetrahydrofolic acid. These two drugs are given together to treat UTIs, bronchitis, traveler's diarrhea, several forms of pneumonia, It can also be administered to treat middle ear infections. Okay, and these drugs are bacteriostatic, so they're just going to inhibit the growth of bacteria so that either another drug or the immune system can actually kill the bacteria causing an infection. This is the end of this lecture. In the next lecture, we'll be looking at antivirals, antifungals, and we'll talk a little bit more about bacterial resistance.
Let's wrap up this video with just a few more questions. Here's the first one. The activity of sulfamethoxazole is due to the structural similarities to, is it tetrahydrofolate, ATP, deoxyguanosine, or para-aminobenzoic acid, aka PABA? PABA. PABA is the answer here. Remember, sulfamethoxazole looks a lot like PABA to the enzymes involved in making tetrahydrofolate. Which of the following is recommended for use against gram-negative bacteria? Would that be bacitracin, polymyxin B, cephalosporin, or polyenes? Polymyxin B. Absolutely correct. Polymyxin B's activity with membranes makes it excessively toxic when taken internally. For that reason, it's mainly a component in skin ointments. What is the target for Cipro? Is that nucleic acid synthesis, cell wall synthesis, protein synthesis, or essential metabolite synthesis? Yes, nucleic acid synthesis. Remember, Cipro targets proteins that are involved in duplicating the bacterial chromosome. So it's something like a magic bullet. Wow, that was a long video, and you made it through. Congratulations! In this video, we've talked about a lot of different drugs. Some of them inhibit protein synthesis, some of them inhibit nucleic acid synthesis, and then we talked about a couple of drugs that inhibit the synthesis of essential metabolite, all in bacteria. Hopefully you took notes on how these different drugs work and maybe some notes about what the targets are as well. That information will be useful to you as you prepare for your next exams. The other thing that I hope you took notes on is how bacteria develop mechanisms to resist the activity of these antibiotics. For example, we mentioned efflux pumps in this video. We mentioned changing the structure of the target for the antibiotic, along with some other things. You want to make sure that you're familiar with those mechanisms of resistance for your exam. That is a big part of your exam. It's also covered in one of your homeworks for this chapter. I hope this video was useful. This is the end of our discussion for bacteria. In the next video, we'll talk about some antifungals and antivirals. I'll see you there.